StatQuest. 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 Hello, and welcome to StatQuest. StatQuest is brought to you by the friendly folks in the genetics department at the University of North Carolina at Chapel Hill. Today, we're going to be talking about principal component analysis, or PCA for short. Let's start off with an example of principal component analysis in action. Here's an example PCA plot that I got from an article that I was just reading. It shows clusters of cell types. This graph was drawn from single-cell RNA sequencing data. There were about 10,000 transcribed genes in each cell, and each dot in this graph represents a single cell and its transcription profile. The general idea is that cells with similar transcription profiles should cluster. And we sort of see that in this graph. We see that blood cells form one cluster that's different from pluripotent cells, which is different from neuronal cells, and dermal or epidermal cells. So the big question is, how does transcription from 10,000 genes get compressed into a single dot on a graph? The answer is PCA. PCA is a method for compressing a lot of data into something that captures the essence of the original data. In this StatQuest, we're going to learn all about how PCA does this compression. Also, we're going to find out what these access labels refer to. Before we dive into the nitty-gritty of PCA, we're going to cover a little background material. We're going to have an introduction to dimensions. Just to warn you, this is going to seem very, very simple. But just hang in there. You'll be glad we did this. It'll keep your head from exploding. If you can remember all the way back to first or second grade, you'll remember that one dimension equals a number line. Now, imagine we had a pretend RNA-seq dataset for a single cell. Here, I've labeled the genes just A, B, and C, and the read counts are 10, 0, and 14 for those genes. We can plot these values on the number line. Just like we did in first or second grade, A with 10 reads gets a dot at 10. Gene B with 0 reads gets a dot at 0. And lastly, gene C with 14 reads gets a dot at 14. If we plotted all genes, we might see something like this, a uniform distribution of transcript counts. Or we might get a non-uniform distribution of transcript counts. Some genes might not be transcribed very much, and they'd be on the left side of our number line, and some genes might get transcribed a lot, and they'd be on the right side of our number line. Even though our number line is a very simple graph, we can get some useful information out of it. Now, let's fast forward to 5th or 6th grade when we learned about two-dimensional graphs. Now we have two axes instead of just one, and now we can plot data from two different cells instead of just one. Here's a pretend RNA sequencing dataset for two single cells. Just like before, we have the same genes, but now we have read counts for two separate cells. If you can remember from 5th or 6th grade, the way we plot the data for gene A is we go over to 10 for cell 1, and we go up to 8 for cell 2, and we put a point there. For gene B, we go over 0 for cell 1, so we don't move at all, and we go up 2 for cell 2. And for gene C, we go over 14 and up 10. If we plotted all of the genes, we might see something that looks like this. Here, we see that the expression in the two cells is correlated, meaning genes that are highly transcribed in cell 1 are also highly transcribed in cell 2, and genes that are lowly transcribed in cell 1 are also lowly transcribed in cell 2. Or we might see that the expression in the two cells is not correlated, meaning if a gene is highly transcribed in cell 1, that doesn't tell us anything about whether it's highly or lowly transcribed in cell 2. Okay, so maybe sometime when we took calculus, we started drawing three-dimensional graphs. That's just a fancy graph that has depth. With three separate axes, we can now plot data from three separate cells. So now our pretend RNA sequencing dataset has data for 3 single cells. And just like before, if we wanted to plot the data for gene A, we would go over to 10 for cell 1, up to 8 for cell 2, and then back 8 for cell 3. We then draw lines perpendicular to each axis to figure out where they all meet, and then we put a dot there. I'm not going to do too many examples of this because you get the idea. So this is what we know about dimensions so far. If we have 1 cell's worth of data, we only need to have a 1-dimensional graph, which is just a number line. If we have data from 2 cells, then we need a 2-dimensional graph, which is just an XY graph that we learned about in 5th grade. If we have data from 3 cells, then we need a 3-dimensional graph. That's a fancy graph with depth. What happens if we have data from 4 separate cells? You guessed it, we need a 4-dimensional graph. The problem is, we can't draw that on paper. And if we had data from 200 individual cells, we'd need a 200-dimensional graph, and there's no way we can draw that. So the question is, are all of those dimensions super important, or are some more important than others? To answer that question, we're going to go back to a data set that just has 2 cells and 2 dimensions. Hypothetically speaking, what if we had 2-cell data that looked like this? Here, we see that almost all of the variation in the data is from left to right. That is to say, cell 1 has some genes that are lowly transcribed and some genes that are highly transcribed. But it looks like all of cell 2 genes are all transcribed at the same level. If we flatten the data, that is, removed the up and down variation, our graph wouldn't look much different from what it looked like before. And if we flatten the data, we could just graph it with a single number line. In this case, we can take 2-dimensional data and display it on a 1-dimensional graph without too much loss of information. Both graphs say the important variation is left to right. Here's another example of how some dimensions are more important than others: TV and movies. TV and movies are almost always 2D. That is, they're shown on flat screens, at home or in the movie theater, and we don't usually have fancy 3D goggles on when we watch them. So they're 2D, even though the subjects in the movie are 3D. This is okay. The third dimension usually doesn't add that much to the story. This is why, when we cough up the extra $3 or $4 to watch a movie in 3D, we're usually disappointed. Anyways, people look like people, things look like things, even when they have no depth and are flattened on a screen. Basically, a movie camera takes 3D information and flattens it to 2D without too much loss of information. To summarize what we know so far, we know that each cell that we sequence adds another dimension, and we also know that some dimensions are more important than others. So what does all this have to do with PCA? Well, PCA takes a dataset with a lot of dimensions, i.e. lots of cells, and flattens it to just 2 or 3 dimensions so we can look at it. It tries to find a meaningful way to flatten the data by focusing on the things that are different between the cells. We're going to talk a lot more about this later. For any biologists out there, this is sort of like flattening a Z-stack of microscope images to make a single 2-dimensional image for publication. So let's start with an example. Again, we'll just start with 2 cells. Here's the data. Like before, the genes are imaginary, so I've just listed them from A to I. And here's a 2D plot from the data from 2 cells. Generally speaking, the dots are spread out along a diagonal line. Another way to think about this is that the maximum variation in the data is between the 2 endpoints of this line. And generally speaking, the dots are also spread out a little above and a little below the first line that we drew. Another way to think about this is that the second largest amount of variation is at the endpoints of this new line that we just drew. If we rotate the whole graph, the 2 lines that we drew make new x and y axes. This makes the left, right, above, and below variation easier to see. We don't have to tilt our head anymore. And like we saw before, the data varies a lot to the left and the right, and the data varies a little up and down. Note, all of the points can be drawn in terms of left and right and up and down, just like any other 2D graph. That is to say, we don't need another line to describe diagonal variation. We've already captured the two directions we can have variation with these two lines. These two new or rotated axes that describe the variation in the data are principal components. Principal component 1, or PC1, the first principal component, is the axis that spans the most variation in the data. PC2, or principal component number 2, is the axis that spans the second most variation. So these are the general ideas we've covered so far. For each gene, we plotted a point based on how many reads were from each cell. Principal component 1 captures the direction where most of the variation is. Principal component 2 captures the direction of the second most variation. What if we had 3 cells? Just like before, principal component 1 would span the direction of the most variation, and principal component 2 would span the direction of the second most variation. However, since we have another direction we can have variation, we need another principal component. That's principal component number 3. It spans the direction of the third most variation. What if we had 4 cells? Principal component 1 would span the direction of the most variation. Principal component 2 would span the direction of the second most variation. Principal component 3 would span the direction of the third most variation. And you guessed it, principal component 4 would span the direction of the fourth most variation. There's a principal component for each dimension, or each cell in the data. If we had 200 cells, we would have 200 principal components. Principal component 200 would span the direction of the 200th most variation. Hooray! Now that we know what PC1 and PC2 are, we know what the X and Y axes are in this figure. PC1 is the direction of the most variation in gene expression, and PC2 is the second most variation in gene expression. But I bet just right now you're asking yourself this question. This is a plot of cells, not genes. How do we plot cells? So far, all we've talked about is how to plot genes. To answer your question, we're going to go back to the original scatter plot for 2 cells. For now, let's focus on principal component 1. The length and direction of PC1 is mostly determined by the circle genes, the genes on the endpoints, or the extreme genes. Now we're just going to move the graph over to the left side of the screen so we can put other interesting things on the right side. If we wanted to, we could score genes based on how much they influenced principal component number 1. And here's a list of qualitative scores that we might give each gene. Genes close to the ends of the line, like A and F, would have high scores because they highly influence PC1. But genes in the middle, like B and C, would have low scores. We could also use quantitative scores for each gene. So genes with little influence on principal component 1 would get values close to 0, and genes with more influence would get numbers further from 0. Genes on opposite ends of the line would get similarly large numbers, but with different signs. So A might get a positive number, like positive 10, and F, because it's all the way at the other end of the line, might get a negative number, like negative 14. Similarly, we could also rank genes and how they influence principal component number 2. Now we have 2 tables of genes and the influence they have on the principal components. One is for principal component 1, and the other table is for principal component number 2. Now that we have these 2 tables for the first 2 principal components, we can use them to plot cells and not just genes. We do that by combining the read counts for all genes in a cell to get a single value. Here's how to do that. First, we return to the original read counts for each cell. We can then calculate a score for cell 1 by taking the read count for gene A and multiplying it by gene A's influence on the principal component, and adding that to the read count for gene B multiplied by the influence of gene B, and doing that for all genes. Here's a concrete example. For cell 1, gene A, we have 10 read counts, and the influence gene A has is 10. So the first part of this summation is 10 times 10. The second part of the summation is the read count for gene B, which is 0, multiplied by the influence gene B has, which is 0.05. We just continue to multiply and sum until we've done it for each gene in the cell. For this example, we might end up with a number like 12. That would be our value for PC1. To calculate a value for principal component 2, we do the same thing as before, except instead of using the weights or the influences on principal component 1, we use the weights or influences on principal component number 2. So in this case, gene A has 10 reads, and we multiply it by 3 because that's the influence gene A has on principal component number 2. We add to that the read counts for gene B multiplied by the influence that gene B has on principal component number 2. In this case, that's 0 times 10. And we just do that for every single gene again. And we end up with a score for principal component number 2, and in this case, that might equal 6. So we've done the math for cell 1. We've got values for principal component number 1 and a value for principal component number 2. Now all we have to do is plot it on a graph. And if we create a graph where the x-axis is principal component 1 and the y-axis is principal component number 2, we can do what we did in 5th grade. We just go over 12 and up 6 and put our dot right there. Now we have to calculate scores for cell number 2. And if we did the math by multiplying the read counts for each gene by the influence that each gene has on the principal component, we might end up with numbers like 2 for principal component 1 and 8 for principal component number 2. Again, we just plot it like we did in 5th grade. If we sequenced a third cell and its transcription was similar to cell 1, it would get scores similar to cell 1's. And as a result, when we plotted it on the graph, cell number 3 would be closer to cell number 1 than it would be to cell number 2. Hooray! At long last, we know how they plotted all of the cells on this graph. These are the general ideas we've covered so far. Genes with the largest variation between cells will have the most influence on the principal components. That is to say, genes highly expressed in some cells and not expressed in others will have a lot of variation and influence on the principal components. The first principal component captures the most variation in the data. The second principal component captures the second most variation in the data. You can use the original data and the first two principal components to get XY values to plot on a figure. Cells with similar transcription patterns will cluster together. And just like they say on TV, but wait, there's more! We can use the graph to identify key genes. Do you see how cells are spread out left and right, above and below? If we wanted to find out which genes had a big influence in putting dermal cells on the left side of the graph and neural cells on the right side, we could look at the influence scores on principal component number 1. And if we wanted to find out which genes help distinguish blood cells from neural and dermal cells, we could look at the influence scores in principal component number 2. But wait, there's even more? Yes, there's a couple diagnostics you can do if you're drawing your own PCA plot. These are ways you can tell if your PCA is actually worth anything. One diagnostic plot is called a scree plot, where you plot how much variation each principal component can account for. What you want to see in this diagnostic plot is that most of the variation is accounted for by the first two principal components. Lastly, here's a terminology alert. The ways I've been describing things has been fairly intuitive, but there's actually a lot of technical jargon for principal component analysis. The numbers that describe the weights for the importance for each gene to principal component 1, I've just been calling influence, or weights. But in PCA terminology, those weights are called loadings. An array of loadings is called an eigenvector. And that's all there is to PCA. So tune in next time for another exciting StatQuest.